Okay, so we'll just go back through the theoretical background behind the exercise you'll be doing today. So that's taxonomical assignment, should be the task today. And yeah, I see there's a lot of overlap between the talks that's been yesterday and today, but I mean, we are going to make profiles of the taxonomic profiles of the samples that we have worked with yesterday and today. And I wrote in the manual that you have been working with that these samples are from human fecal samples, baby fecal samples. So it's we're looking at the microbiota in the human gut of babies. And most of you know that we have different microbiotas or different taxonomic profiles on different sites of the body, if you like. And it also changes over time. So we have a completely different microbiome when we are babies than when we are grown-ups, and it changes all through our life. This is actually not right. We are not sterile when we are, even when we are inside the uterus. Okay, so human microbiomes are quite straightforward to study. There's lots of studies on that, but there are other studies which we find more interesting. We are working with marine samples, and we heard talks from soil samples and so on. And this one I found particularly amusing, where they have analyzed the windshield of a car so they've looked at the taxonomic profiles by taking a car and driving two different distances in the United States, and they have sampled what was on the windshield of the car from these two different trips, and they've made a taxonomic profile of that, where they've looked at the relative abundances of the different, well, all the way from phylum and down to genus here. And for those who have good eyes, you may see that they also find some Homo sapiens in the samples, but they promised that they didn't hit anything on the road. There must have been some other kind of contaminations going on there. So lots of different environments to explore. So yeah, you've been through this, so I won't go in detail, but this was the old way of studying different microbiomes, that we had to grow them. And as you know, especially from the environment, very few species are actually growing, maybe less than 1% in some of the environmental samples. So now we do direct isolation of DNA, and we sequence it, and theoretically we can cover everything that's in the sample, theoretically. So what we are going to do today, so on Monday the whole day was dedicated to amplicon sequencing, looking at 16S ribosomal RNA genes. So what we will do, we will focus on this part, the random sequencing. But I'm just showing both of them just so you have a clear picture of the differences between those. So as you know, with amplicon, you're amplifying parts of the ribosomal RNA gene. Random sequencing, we're just sequencing everything. And what the main differences are is that when you do amplicon sequencing, you can mainly look at, well, you are just looking at taxonomy, really. While when you do random sequencing, you can look at the taxonomy, but you can also look at the functional potential of a sample. So that's the main difference. I think Espen will talk more about the functional part later. Some other differences I've put up here. So when you do amplicon sequencing, you use specific primers to amplify the 16S, and and so on. But down here where we are at the data analysis part, what this course is about, so you do quality control, filtering, and taxonomical profiling here. When you do random sequencing, we do almost the same steps, but since we have all kinds of genes in here, we need to identify the 16S ribosomal RNA, the reads that contain those, and then actually do pretty much the same as in this pipeline. So this is what you're going to do today. So yesterday you did quality control and filtering. You also did assembly. But today you will continue with the filtered sequences and try to identify 16S ribosomal RNA and annotate them against 16S databases and make a taxonomic profile. So that's sort of one of the exercises today. So I put up a slide showing some pros and cons between 16S and amplicon. We are mainly doing random sequencing, so I'm very pro random sequencing, but I mean both methods have their pros and cons. So yeah, so I mean when you do 16S amplicon sequencing, I mean one of the disadvantages is, as we've heard earlier today, that these the primers that you use aren't universal. You won't see everything that's in the sample, so you may miss out on certain taxonomic groups that you aren't able to amplify. Yeah, if you look at the expenses, it's still cheaper with amplicon sequencing, and also if you look at the computational resources, so the virtual machines that you're working with now are a little bit more powerful than a normal laptop, and we are working on quite small samples now and few data sets. If you're working with larger data sets and let's say hundreds of them, then that this could be a limiting resource, the computational resources. So it's requiring more if you do random sequencing. Yeah, and this is also a little bit interesting. So if you're studying if you're working with random sequencing, you can also look at viruses and eukaryotic species from the same sample together with the bacteria. Yeah.
So I have a question related to that, because was it mentioned that the metagenomic data has to be filtered for RNA sequences as well?
Yeah, I mean, to obtain a better, for example, to obtain a better assembly, we normally filter out ribosomal RNA before the assembly. That's quite common to do.
Those are from DNA sequences, right?
Yeah.
So you would not get sequences from actual RNA. But I'm just thinking that if you have RNA viruses in your sample, you will not get sequences of those in the actual data, right?
No.
What kind of data do you mean, like the assembly or the?
Just metagenomic sequences. You will not see the RNA viruses in the data at all.
Probably not, I guess. Depends.
No. No, you need to extract RNA also. You're right.
There's no method to sequence DNA and RNA.
No, you need to reverse transcribe it and make the library out of that. Probably similar to transcriptomic methods, I guess. Yeah, I have no experience with those kind of samples. Okay. Yeah. So what you will also see today, we will work with something called lowest common ancestor tool. So, just very briefly, what that is, if you have two similar reads, which the tool cannot place into, let's say, on a genus level, it can separate if it belongs to genus A or B. Then it moves it up to a higher level in the hierarchies because it's too similar. maybe it's 99% similar to, I can't read, it's too small, but let's say A and B, so it doesn't know where to put it. So it will put the read, the 16S read, one level up in the taxonomic hierarchy. So that's how the lowest common ancestor works in principle, and that you will see that you will have some reads that are mapping all the way out down to the genus level, and some that are maybe all you cannot separate more than the order or even the class level. Yeah. Some other. So this is a paper by Joel from last year where they were comparing both the methods and tools. So they were comparing amplicon versus random sequencing, and as mentioned several times, so the amplification step leads to biases, and there are actually some. So they made a mock culture of a little over 10 species, and what they saw was using the most common set of 16S primers. What they saw was that they got an overrepresentation of some of the families in the sample because the primers were perfectly complementary to the genome. So, for example, Clostridium and Lactobacillus was overrepresented, and other bacteria were not because they weren't the primers didn't match that well. So it's nice to know that, and I'll come back to that in a second. But the reason why I'm just telling you this is because you're going to do two different methods today. You're going to extract the 16S, look at the taxonomic profile, but you're also going to use another method where you look at other genes than 16S from the random sequencing. So you'll see some differences in choice of methods too. Well, this is from the same study, just showing. So they claim that random sequencing, so their name is shotgun, is more. It gives better results than 16S. It's closer to the real mock culture that they had than 16S. But these are from 11 species that they were looking at. Yeah, so there are differences there. Okay, I'm just gonna go to yeah. So this is similar to a slide I showed earlier, only that you're also going to. So this is 16S we are looking at in the random sequencing sample that you have, but there are also two other methods we can use. One of them we heard about earlier today, binning of contigs and producing a profile from that. We're not going to do that in the exercise, but we are going to use this method here, which is called, well, we look for clade-specific markers using a tool called Metaflan2, which Rob mentioned earlier. It's a really fast method suitable for human samples from the human microbiota, not necessarily from the sea or the marine environment or the soil. So you're going to make two taxonomic profiles. One extracting the 16S reads and one looking at the clade-specific markers. Okay, so this is actually an overview of what you are going to do today with the tools in black boxes here and yeah, the process here. So you have done the quality control and trimming, and you're not going to do. Okay, so this slide is a little bit old. You're not going to remove contamination. The reason why we skip this step is it takes quite a long time to run this tool to remove contamination. So this is samples from human fecal. So in some samples we have seen human contamination up to 90%. So 90% of the DNA that we have sequenced is from human. So, but in these samples we selected from for these exercises, it's much less than 1% in it. So we decided to skip this step. But it's nice to know that there are tools that can remove contamination from a host, for example. So we have also worked with sample from the salmon, fecal, from the, gut samples from the salmon, where we have screened for salmon DNA and removed contamination. So this step you're not going to do. So what you're going to do is to take the trimmed reads, and you're going to run some tools to identify all reads with 16S ribosomal RNA sequences in. And you're going to use a modified version of the Silva database to annotate all the 16S reads, and then make a taxonomic profile. And yeah, we have a visualization step at the end also, using Krona tools, which Rob showed a lot of slides on earlier. Just gonna show you that there are different tools to predict ribosomal RNAs in a sample. So we are going to use the RNA selector. We have done some benchmarking, just showing that it's performing better. So it's faster than other tools that we have compared against. So that's the reason why we chose that. But the main, what will influence the results more than the choice of a tool is the database. And yeah, again, I'm repeating what's been mentioned earlier, but the different databases they have really a big impact on the results. So you're going to use the Silva database here, and not RDP or the green genes. So as long as you stick to one of them, you're able to compare the results you have against each other, relative to each other. So, okay, yeah, there's lots of tools available to do the profiling. Tomorrow you will use this tool called Megan to look at the taxonomic profile, compare things. Okay, so this is the second. So now moving on to the clade-specific method, clade-specific marker method to make taxonomic profiles of your sample. So we're not looking at 16S anymore. So this method is that you map all your reads, all the trimmed reads, against a database which is quite small, the database, but it contains genes or fragments of genes that are specific to specific clades in the sort of in the hierarchy. And then yeah, so the tool you're going to use is Metaflam2, and it includes 1 million of these markers or fragments of genes from 7,500 species. Now I've tested this tool on environmental samples, and it doesn't find anything there. So it is mainly for analyzing samples from the human microbiota. Okay, yeah, just a quick sketch on how this works. So, what you have are, so if you imagine 3 different clades, and they have and there are different species within those clades. So this species here, A1 in clade 1, has 2 genes, and B has 1 and 2 genes. But all of these blue genes are specific to clade 1. You'll find them in some of them. And similar here, the green ones are specific to clade 2 and the red ones to clade 3. So when you do random sequencing and you start to map these, or Metaflam maps your reads onto these clade-specific genes, it will both look at the coverage, so how many reads that maps to particular clades, and yeah, which clade it maps to, and then produce a taxonomic profile showing the relative abundance, how how much of your sample, how many percentage of your sample that belongs to clade 1, 2, and 3, and so on. So this is what you're going to do in the second part of the exercise today. Yes, binning was thoroughly explained earlier today, so, and we're not going to do that, so I'm just gonna skip this one. Yeah, and just finally here, I'm just gonna say something about sequencing depth. So there's a lot of studies that has sort of looked at how much you actually need to sequence it, but in order, I mean, it's always a question of how much money you have, but studies show that you don't really need to go that deep to get most of what's in the sample. It's those special ones you need to go really deep to find. So yeah, I mean, so we are not. I mean, we are working with samples that are not that diverse, and we think we cover most of what's in the sample. I think yeah, it will be exciting to see how many different taxonomic groups you find with the two different methods, and see. Okay, so today so you will continue on the MetaQuast. Look at the MetaQuast output now. It should be done for most of you. But once you have done with that, we continue on exercise day 3. So you need to enter the day 3 folder in your on your virtual machine, and then you will find in the raw data folder you will find the trim reads, and you can continue from there. So there's 2 exercises you do. One where you annotate the 16S sequences, and the other one where you look for clade-specific markers using Metaplan 2. And in the end, you will make a nice visual representation of the taxonomic profile from both of the methods. Okay, so I think we'll continue until a little over 3 o'clock, and then Espen will give a short introduction to the functional part of the exercise, where you're going to annotate the assembled genome that you produced yesterday.